we have speaker Dr. Animesh Samanta from Srinagar University. Speaker has requested me to keep it short about his introduction, so I will make it short on his request. He is very well known to me as we were batchmates since college and in our master degree also, and um, almost more than 1.5 decades we are known to each other academically. Our speaker, Dr. Animesh Samanta, did MSc in chemistry from IIT Madras and PhD from National University of Singapore, NUS. After his PhD, he did his postdoctoral research in Singapore in NUS and another company which is named as A Star. After his postdoctoral research, he joined um, as an assistant professor in 2016 as a Ramanujan Fellow at CSR in IIST Trivandrum. After that, he joined Srinagar University uh, in India. He has uh, more than 37 publications in reputed journals and also 2 patents. One of them got licensed to also. He is also the recipient of first prize of JJSI Outstanding Best Graduate Thesis Award in Biotech. Let's listen to him about the scientific story on nanotechnology in healthcare. Uh, Dr. Samanta, I would request you to inform.
Thank you so much, Dr. Shaha, for kind word of introduction. And it's my great pleasure to be here, especially from the invitation from my long time colleague. And also he's one of my kind of respectable dada, we call them, because of his gentle behavior. So I'm very much happy and pleased to be here. First of all, I would like to thanks to the PEC, Akal University, and the HOD for giving me opportunity for sharing my thoughts on the topic of nanotechnology in healthcare. So before I go to my talks, so I first would like to welcome to all of you to Srinagar University, which is very close to Delhi. By chance, if you visit Delhi, so just let me know. I will heartily welcome to our Srinagar University campus. Now, I will talk about my topic today. So that is how nanotechnology can be useful tool for diagnosis and therapeutic. So I have seen few of the webinar from Akal University, especially on 22nd September, presented by Dr. Gaurav Prakash regarding the blood cancer, which during that time, I realized this topic could be continuations of that topic. Why? That was from that doctor point of view, it was a clinical purpose. Now here, as a researcher, as a chemist, we wanted to give the highlight on preclinical analysis. And in that particular cases, how nanotechnology play the key roles that I wanted to highlight for today's talk. So first I will introduce about nanotechnology and the applications in hepatotoxicity, followed by cancer diagnostic. So by using nanoparticle, it can be considered as a SaaS-based technology, MRI technology for diagnosis purpose. Not only diagnosis, nanoparticle can be useful tool for therapeutics. So this therapeutics purpose, we can consider about surgery, photodynamic chemotherapy. During that time, drug delivery is a key important. That will be highlighted. But now it is even advanced research is photodynamic therapy or thermal therapy, magnetic vortex, how it can be useful tool for cancer therapy that will be highlighted. And at the end, this diagnosis as well as the therapy combined together, we call them as a theranostics. So that also will be given some examples. First of all, what is nanotechnology? So before talk about this nanotechnology, the nanoparticles or like a colloidal gold particle, it was synthesized Sigmundi 1925 and got the Nobel Prize because of the development of a colloids in 1925. But nanotechnology, this is the concept was proposed by Nobel laureate Richard Feynman in 1959. Later, the word redefined practically by Professor Taniguchi in 1974. According to his opinion, that nanotechnology mainly consists of the processing of a separation, consolidation, and deformations of a materials by one atom or one molecule at the nanoscale. That nanoscale means the variations is 1 to 100 nanometers. So that's why whenever something is a nano, then its optical property and other property is too much variable from the bulk property. As you can see from here, the bulk property is a reflections of the optical light. However, the nanoparticle from the bulk to nano. Whenever it is a nano, the gravitational force does not applicable. It's a wave particle duality in nature. There is a quantum confinement. Previously, bulk electrons are delocalized, but they are confined and increase surface to volume ratio. That's why that nanoparticle play a big attention for applications in multiple field. So here I'm talking about nanobiotechnology. When the nanotechnology we wanted to apply in the healthcare, societal benefits, then we define them as a nanobiotechnology. So it's a truly interdisciplinary research. So it's combination of nanotechnology and as well as the biotechnology. So the nanotechnology also is a kind of physics, chemistry, and material science. Similarly, biotechnology can be considered about bioengineering, biochemistry, and microbiology. So it's a true interdisciplinary field where this nanotechnology play a major role for big value addition to the societal life. So applications of this nanoparticle, if we can think about in a solar system, like huge amount of applications. However, today, I am going to focus mainly on the healthcare, biomedical, drug delivery, antibacterial or UV protections, like that particular directions, and my focus will be on that particular directions. So before, maybe there are few students, they might be not much familiar about nanoparticles. So to redefine, what is the size supposed to be? Like if we think about our hair, one hair is approximately size is 60 to 80 micrometer. Now we are talking about at nanometer range. That means it will be much, much smaller with respect to even our hair. So one glucose molecule could be in angstrom unit, then one protein could be in 1 nanometer, and one nucleic acid could be 1 to 10 nanometer. Virus could be 100 nanometer, but one cell could be like a 20 micrometer. So we are actually handling in this range 1 to 100 nanometer. So few examples are like micelles, liposome, dendrimer, and we'll discuss more about details about these nanoparticles. So they are divided into two parts. We can consider them as organic nanoparticle. Organic nanoparticles are made of carbon, hydrogen, nitrogen, oxygen, majorly. And these are like micelles, liposome, nanogel, and dendrimer. There are inorganic nanoparticles also. Inorganic nanoparticles are made of a different metal. So when we are using a iron, that means it will carry the iron property, that is a magnetism property. So this can be considered as a magnetic resonance imaging, MRI contrast agent. Like similar ways, depending on the metal, it can be shows a different property. So that's why it is a very much attractive and for applications and for research purpose. So obvious, being a chemist, what is our main aim supposed to be? How we could able to synthesize such a small tiny nanoparticles. There are two approaches. One approach, we call them as a top-down approach. Another is called bottom-up approach. So bottom-up approach is nothing but as a chemicals method. If something is a chemical method, that means that is a chemistry play the key roles. And when we say top-down method, so in this bottom-up approach, actually you need to have some atoms. So multiple atoms combine together to form a cluster, and multiple cluster can combine in nanostructure. Like the way we can think about one brick, we take a few hundred or thousands of bricks to make a one house. So that's the way we could able to say it is a bottom-up approach. And the another way, like if mechanical engineer, what they will do a bulk things. They will try to grind small, then can make a nanostructure. So that is a physical method. So like the way it can be made it as a house from the mountain itself. So these two approaches. So here I will give you a quick video by which we can show like how we can synthesize this kind of nanoparticle in a very short time. So here the basically simple aqueous solution was taken, and this aqueous solution can be added Au3 plus. So this Au3 plus, now it has to be reduced to Au0. Then only we can form it's a atom, it's a gold atom, and then multiple number of gold atom can form like a cluster to form nanoparticles within a few minutes. You can see 2 to 10 minutes within that time period how the color is changing because of the formations of a nanoparticle in presence of some reducing agents. So this reducing agent can be considered simple chili powder, or can be considered as citric acid, or any chemical reagent. So by which we can actually within a few short time we could able to synthesize this kind of nanoparticle. That's actually beautiful of synthesizing nanoparticles. Where we wanted to synthesize one natural product, it takes really long time, maybe 50 to 100 steps. So with respect to synthesizing nanoparticle is much easier and faster. So this nanoparticle can be tuned their shape. So Professor Wanasio in Science 2012, he first published about shape control synthesis of a gold and silver nanoparticle. So shape, it can be like a spherical in shape, either it can be a star-like shape, sometimes it can be like a rod-like and hexagonal and diamond-like, whatever we want. So the chemistry, they can tune the chemical condition and the surfactant by which they could able to synthesize whatever the structure they want. Like a one jewelry, so they can make jewelry whatever they want the design. Accordingly, chemists also can do at nano level. All the sizes maybe 20 to 50 to maximum 100 nanometer. So that can be considered as an inorganic nanoparticle. Also can be considered like a organic carbon-based nanoparticle, like a single one carbon nanotube, multi-wall carbon nanotube, and some nanowire can be synthesized. So this kind of nanoparticles can be considered your chemical toolbox. And this chemical toolbox can be used further for different diagnosis as well as therapeutic purpose. So this kind of nanoparticle, why it is very important? Because we can actually tune their optical property. How can we tune the optical property? So here we can see there is a 5 nanometer to 100 nanometer, their optical property is changing. The similar way, silver nanoparticle also their optical property can be tuned. Similarly, there is a gold nanorod. So this gold nanorod also their optical property can be changed based on their aspect ratio. So the aspect ratio is actually length and width can be tuned based on the reducing agent and the surfactant. So as we can able to tune the optical property, it will be really a huge advantage for different applications. So this kind of nanoparticles are, when it is a bare nanoparticle, it's always a big issue considering the toxicity, because we mentioned it is a high volume of the surface. So it can react with a different cellular organisms. So that's why we need to make them some coating materials. So this coating material can be used polymer, can be used biopolymer, like a carbohydrate, protein, nucleic acids. So because of the like a coating of this kind of protein, nucleic acids, then they became biocompatible. So this biocompatible nanoparticle can be further modified on the surface. So now nanoparticle carry their own optical property or their magnetic property, and then the surface can be further modified with targeting antibody, and then wherever we, or even it can be loaded with a different drugs, then it can actually target to the right location. So can be considered a targeted delivery. We'll see some more examples in few slides. So that's the advantage of having nanoparticles with a high surface volume. So that's why they have a very huge application, especially like the percentage of publications nowadays. It's majorly they cover in cancer research, followed by inflammatory, cardiovascular, and some other diseases. But why, of course, the question is why the more research ongoing on cancer based on the nanomaterials. There is some good correlations between nanoparticles and the tumor tissue. That will be discussed another 1 to few slides later. So not only like a healthcare, these nanoparticles have been extensively used in home, automotive industry, food beverage, in different applications. However, for today's topic, we majorly covered about this nanoparticle in healthcare applications. So now, as we mentioned today, our main aim was like nanotechnology for diagnosis as well as the therapeutic purpose. So how we can use this nanoparticle as a good toolbox for diagnosis. Like whenever we have some symptoms of a disease, what do? We go to the pathological lab. And then we provide the blood sample, urine sample, or saliva. And it was after analysis, just we got a report, but we never know what is going in between. So that's nothing but a different kind of test. One example, if I can show you here, like a P24 overexpressed red blood cells, which is indications of a HIV. So by using simple like this device, there is an antibody will be loaded on the trip. And then whenever this blood sample will be passed through it, then if this P24 will be recognized, then they will give us some positive signal. As this red blood cell itself is a color, then we may see like this kind of 2 red color spot, which is a indications of a positive HIV test. Using the same concept, nowadays, whenever we open different kind of news channel, newspaper, we always can read like a COVID-19 test can be done by using simple paper-based technology. So here, maybe simple paper is there, it's lateral flow assay. So in that kind of cases, what is our sample collected? We collect saliva sample, which does not have a color. Then how can we able to detect them? So for that purpose, nanoparticle play the role. So the nanoparticle usually conjugated with the antibody, and this antibody can recognize the antigen having SARS-CoV-2. And then after that, if you can pass through it, if there is a positive, then the gold nanoparticles actually will be remaining on this strip. So that's how you can able to see the color. If it is a color, then it's a positive test. If it is no color in the test, that means it's a negative test. So basically this color is coming from, it's a gold nanoparticle. So by using this concept, there is a low cost antibody-based diagnostics kit are available for COVID detections. And another method is a PCR method, which is relatively expensive method. So that's why the easy method can be developed by using nanotechnology. Now the question about we are talking about only extracellular matrix, which is a urine sample and other sample or blood. But if it is something inside our system, that is we call as a in vivo. So during that time, what we'll do? Usually what we do, endoscopy in clinical trial, colonoscopy, MRI, CT scan, by which we can able to say about what is the disease conditions of normality, abnormality. So for that kind of cases, actually we need a different kind of contrast agent to make it better. So always much easier by using urine or blood sample, but when if it is something like maybe some liver got damaged, how to detect it? So this liver damage detection can be done by help of different small animal to big animal. So by using this, that's why usually takes a little longer time, because we need to have some preclinical analysis. Being a researcher and today being a students or faculties, so their first aim to do this kind of things in a laboratory level as a research. And after all kind of things can be passed, then only it can goes to the clinical level. So today I will give you this one example of that. So in that case, how we do the diagnosis? By using a different contrast agent. Simple one example I wanted to show to you. Let's see, this is a sugar solution and there some dye is added. So now the ants drink it, and after that we can visualize, if we go through in microscopic level, where the sugar moiety goes, we can able to visualize them. So especially what particular cells or cellular organelles can be specifically identified. Sometimes we may not able to see in naked eye where is the locations of the tumor, but it is possible to detect by imaging modality to see the tumor locations. How does it really happen? At molecular level, what happened? We need to have some contrast agents. What is the contrast agents? That I'll discuss later. So there is electromagnetic radiation. All of you aware about it, like a radio wave to gamma ray, which interact with this contrast agent. And after that, they will give some output signal. Either can be scattering, emission, and complementary we can get to visualize them by using detector or naked eye. So this kind of contrast agents can be organic small molecule. So now my current research is basically organic small molecule. Why? Because of the efficiency of instruments. So availability of instrument. However, my previous journey was in nanoparticles. So here like a quantum dot is the one kind of nanoparticle. So to make it biocompatible, these are the different kind of protein are conjugated. Some upconversion nanoparticle, sometimes can be a hybrid system. So there is a, all of you may heard about Raman spectroscopy, which is a discovery by C.V. Raman, but application towards in diagnostic was not that much familiar because of the signal intensity was very poor. However, that problem has been overcome in 1976, and there is an enhancement happen when we use some nano substrate. That means it can increase the intensity maybe 10 to the power 7 to 9 orders. Now it's become a very good platform for diagnostic purpose also. So we'll see some example for today's presentation. So different kind of this kind of contrast agent, if we can develop them and if we check their biocompatibility, then we can use either of them, optical, ultrasound, positron emission tomography. These are the different modality by which we can do the image analysis for early diagnosis of disease, including cancer and hepatotoxicity and other. So basically to do this also it takes really time. So here at the first you need to develop a different nano-based technology or any other contrast agent. Then you need to go for preclinical development, followed by if everything passes nicely, then you can go to the clinical development. So a doctor usually suggests something which is already clinically developed, but researcher, we do lot of research to test their initial viability. So that is actually takes really time. So here one of the example I wanted to give you, like a hepatotoxicity. As I mentioned, hepatotoxicity means it's a kind of liver disease. So why this liver disease happen? Just you. So now we say about COVID-19, it's kind of fever. So the one of the most effective medicine, we can say paracetamol. So this paracetamol, if we use excess, then there should have some side effect. Or like people love to drink like beer, alcohol. This alcohol actually cause for dehydrations of a liver. So that cause of the liver toxicity, and this liver toxicity can be monitored by using nanotechnology. So here we have developed previously during my postdoc that one nanoparticle based nanoprobe which can be monitored like a amount of paracetamol, how does it affect to the liver? How does it work? So basically the paracetamol, if it is excess, then it can form an NAPQI as a byproduct, and this NAPQI actually reacting with the tissue and tissue got damaged. At the same time, they give a byproduct as peroxynitrile. So this peroxynitrile, if you can develop some probe which can detect this one, basically from there we could able to detect the paracetamol concentration. By using this concept, what we tried, we just tried a one organic molecule, another inorganic complex, and then combine together. This is initially, it does not give any color. But whenever we treat paracetamol, we generate a peroxynitrile, and this peroxynitrile will give you the color. Basically, we wanted to see the color. So that is actually tested in animal model. So in the animal model, it is very difficult to see where is the liver, but directly you can do the image and you can understand this is the position the liver located. And when we collected, of course, it is found in the liver. So now treatment of a different amount of paracetamol, so like 250, 500 milligram per kg, then we can see there is a drastic effect on the liver damage. That means we could able to monitor the liver damage by using this kind of nano hybrid systems. So that's the one example I give you. And then we'll talk about nanoparticles are majorly used, as we have seen more than 42% research on cancer. So we'll talk about cancer research. So previously, as Dr. Prakash has already have described very beautifully, like what is the cause of, why the importance of a cancer. Still, to briefly describe today, like a cancer is the definitely it's a major cause of death. And to develop a drug, like there is a chemotherapy available, but to develop a drug, it really takes a long time, maybe 12 to 20 years. So after that also, there are different source of cancer. So maybe it is not universal drug. Maybe it is effective to breast, but may not effective to lung. That's why. So we need to have a early detection. So father of modern chemotherapy, Sidney Fever, have mentioned like the detection is the key way we could able to recover the cancer on right time. So detection, how it can be done. So that we'll talk about today. So now basically what is cancer, if we just briefly talk about it, obviously Professor Dr. Prakash already has mentioned, it's not a single disease, a process of a uncontrolled excessive growth of a cells. That's called a malignant tumor. It's caused because of the different kind of environment, our dietary system, all these things. What happened at the end? Actually, it happened that DNA got damaged. Always there is a DNA repair mechanism, but excessive DNA damage does not control by the repair mechanism. So that's why there is a mutation in the gene. So because of the mutations of the gene, they will start modify the gene, and some of the gene cause for the excessive growth. So because of this excessive growth, basically you can see that from this is the primary one. However, it start growing and it's become a very big tumor. But this is the stage actually we able to detect it. However, our aim is not. We wanted to detect at this beginning. If we could able to detect, the diagnosis has to be more efficient way. So for that purpose, nanotechnology could be the best solution for current scenario. So what are the available technologies already available? Yes, there is a available of a biopsy. We can cut some tissue. It's a painful, similar way endoscopy. Also some drawbacks are there. There is pap smear for cervical cancer detections. And these kind of things eventually like a might be a little late stage. With respect to that, circulating tumor cell detections by using nano device, this is the most advanced. I will talk about how it can be worked by using nanoparticle. So as I mentioned, circulating tumor cells. So circulating tumor cells, what happen? Actually, there is a kind of primary tumor. Maybe it's first started at the beginning, maybe in the breast. However, it may not be remaining on the breast. It can actually try to, there is a epithelial mesenchymal transition can be happen, this EMT. So from one cell, they start to grow to the blood vessels. And these blood vessels, then again, they start to move to some other direction. That's called as a process of a metastasis. So metastasis is the key cause. 90% of death in cancer caused by metastasis. So then maybe we are treating one cancer, but now it is going to the secondary tumor. So that's why uncontrolled growth is very difficult to treat them. So for that purpose, the CTC detection is very important. So how we can use the nano platform? So the nanoparticle, as I mentioned, their surface volume is very high. Now we can conjugate it with the reporter molecule, which can give a Raman signal, and further modification to make them biocompatible, and you conjugate it with antibody. And this antibody can target to the antigens having on CTC. So here the whole blood can be centrifuged to remove the red blood cells, And these white blood cells, along with circulating tumor cells, can be detected by using surface enhanced Raman scattering technology. There is another technology currently used, that is actually nano platform. So here magnetic beads. So these magnetic beads conjugated with the CD45 antibody, which can target to the white blood cells. And here, because of the size, RBC can be excluded at the beginning. Because of the magnetic beads, that's the nanoparticle, which is CD45, that antibody target to the white blood cells. And now here is the applied magnetic field. Because of the applied magnetic field, what happened? Then they are attracted because there is a nanoparticles of magnet. So they will attracted towards this field and circulating tumor cell can be separated. Usually circulating tumor cells, whenever it is in the blood, white blood cell always try to kill them. However, some of them survive. So because of that, the concentrations of CTC are always low to detect it. But this detection can be detected by using this nano platform. So that's the advantage of having nanotechnology for cancer detection in the blood level. But if you wanted to do something in vivo, means which is within the body, something happened, we wanted to detect it. How we can do it? For that purpose, as I mentioned, we need a help of a medical, some small animal, and we need a different kind of imaging as well as therapeutic process. So in that kind of cases, nanoparticle play a vital role. Just few minutes back I mentioned nanoparticle and the cancer or tumor has a very well connection. What is that connection here? When we talk about a normal blood vessels, there is no kind of leakage. It's a very normal. But wherever the tumors start to grow, their blood vessel is a leaky vascular nature. You can see this kind of leak. So that size also perfectly matching 10 to 100 nanometer. That means our nanoparticle is also 10 to 100 nanometer. That means it easily can pass through in this kind of gate. But if we think about a small molecule, it's a very big with respect to them. It can goes and then again it can be comes out. So that's why nanoparticle can be used as a very good tool for targeting cancer. So there is a two way. One can be passive targeting, another is active targeting, especially in the passive targeting. So these are the nanoparticles, they start to go to the tumor locations and accumulate it more and more. But for the active targeting case, there is a tumor. On the tumor, there is a some receptor. And this receptor can be targeting with the antibody. And this antibody, whenever they recognize, then they can actually go inside the cell. So by these two ways, nanoparticle can be delivered more efficiently to the tumor location rather having to the normal tissue. So by using this concept, actually, we could able to develop a different technology or diagnostic therapeutic device. So basically, any kind of tumor tissue, if you can analyze them, basically, as I mentioned, some of the cases, if we use the cancer drug, there are different cancer drugs already available. Sometimes they do not stay in the tumor cells. It's actually excreted from the cell itself. There is a heterogeneity and some cases the permeability in the blood, because whenever we inject something in the blood, then it has to stay a little longer time in the blood vessels. Then slowly it can be uptaken. But if it is not stay for a long time, what happens? It can be excreted from the body. That means actually we are losing our drug. So the retention permeability for nanoparticle is relatively high. That's the reason nanoparticle can be considered as a good tool for targeting cancer research. So here one example I wanted to give you. It's like a source-based Raman, as we mentioned, Raman intensity was weak, but now it has been used like a well-defined some nano composite has designed and synthesized. And then this the antibody, they can target to the cancer's antigens. So after targeting, now this is the actually beautifully construct. So if it is happening to the cell at in vitro level, there is a possibility also in vivo level. That's why this tumor, it can target and they can able to see the signal. In the same direction, also we try to do it. In that kind of cases, we develop a more efficient Raman reporter, and this Raman reporter we conjugated with some protein to make it as a biocompatible, and after formations of this biocompatible particle, then we target to the tumor, and we could able to see the image. It's a positive image representing at molecular level. Maybe we can see this is a tumor, but what tumor it is, we may not know. But because of this analysis, we could able to say this is a xenograft of breast cancer. So now we are going to give you one comparison. The comparison between like a two different technology. So one is optical, it's a fluorescent technology, another is magnetic based technology. And here also we are going to detect some metastasis tumor. What we mention about metastasis, like there is a primary tumor, that primary tumor, some of the cells will migrate it and it will go to the some other organs. And when it goes to the other organ, then can we able to detect each locations of the tumor by using nanotechnology? Yes, it is possible. So here, one of the kind of optical nanoprobe has synthesized, another MR nanoprobe has magnetic resonance imaging nanoprobe. It's a gadolinium based, and then they mix together, and then let's see, this is the primary tumor. So this primary tumor, after 4 weeks, they actually dislocated to different area. Now, this one, by using these two contrast agent, then they can image it and they can analyze them, and it can be easily visualized where the location it is. So with respect to the optical imaging seems to be much more promising than the MRI imaging. However, this MRI imaging is already in the clinical process, and always we do MRI scan. However, this optical imaging is still in the research field. So hopefully it will come after few years in the clinical purpose. So which have a much better sensitivity. That means we are hoping for early diagnosis. So far we try to see different example of kind of diagnosis by using nanotechnology. So what is the timeline? What is the hope we have? So we can see 1895 first X-ray, followed by some chemotherapy 1942, then PTFAT, then first coming as a initiation of MRI contrast agents. As we are talking about, different contrast agents are developed, and these contrast agents are further modified to make it more effective, and we are further progressing in different kind of carbon dots, also organic nanoparticles. So there are different nanoparticles are in a phase 3 or final trial, which may come up to the real clinical life. So that's the actually hope, and that's the actually history about it. So now we'll talk about, so we understand about the diagnosis. Now we'll talk about some about therapeutics. So when we diagnose them, now next question is, how do you do therapy? There is a options of a surgery. Obviously, we care about it because of so many things. And radiotherapy, it's also really harmful because it's a strong radiation are applied. Chemotherapy, which also has some lot of side effect. You may have heard about it. Or Chuburaj, who already like became much better conditions. Now he's recovered from the cancer, but there is a side effect of hair loss or some other health issue. So alternative, we are thinking like all researcher, they are thinking, is there any possibility without introducing any drugs, can be possible to detect it? Yes, that is also possible. Like whatever in our body having water and there is a oxygen is dissolved. And this oxygen, if you can make it as a triplet to singlet oxygen by triggering light or any way by using photosensitizer, then singlet oxygen is definitely a toxic for the cellular condition. So they are very reactive and they can react and further they can destroy the cells. So that's kind of research is ongoing nowadays. We'll see some of the example here. As I mentioned, surgery for treatment of cancer. So in that kind of cases, like the diagnosis first we do, and then before do that surgery, during that time what we need to have some nanocomposites. So this nanocomposite can go to the tumor locations. When it goes to the tumor locations, then we could able to visualize the tumor where it is after scanning, and then it can be excreted. And sometimes there will be metastasis. So it might not be so easy to understand, but after imaging, we can say it's a interstitial or peritoneal locations tumor it is. So there is the chemotherapy, as I mentioned, there are different drugs available. Depending on the cancer, there are different drugs available. However, this kind of drug, if we see the structure, being a chemist, we always think about some of them are very hydrophobic in nature. If something is hydrophobic in nature, doctor will say it is not very difficult, it is not a painful, we can just use a saline. But if it is not water soluble, how it can go to saline? For that purpose, we need to do formulation. So this formulation, we need to use different nanoparticles, and these nanoparticles formulated them. Like this is the drug which is hydrophobic in nature, and we can actually incorporate it within the micelles liposomes. These are actually now commercially available in the market. So even similarly, inorganic nanoparticle, now you can treat them. Then it can goes to the right locations through passive nature. It can goes to there, and then we could able to detect it. So depending on the size, their morphology, we can actually control them. Like a very small size, it's not good, it will go to the kidney, it will stay more to the liver, but size 10 to 100 nanometer, it's very effective for targeting to the cancer. So that's why like our other organelles should not be affected, but it should be affected only tumor, depending on the surface modification, depending on the size and shape. That kind of research is ongoing extensively, and many of them are in a good condition to go for phase 2 and 3. So to summarize this one, I just wanted to give you a quick small video which can give you some idea what I said that will be reflected here. So let's this one.
The particle formulates in an organic chemotherapeutic agent, while the white strands represent diblock copolymers, which are realistically more flexible than shown here. A diblock copolymer is composed of two joined polymer chains, a hydrophilic or water-loving block, shown in blue, and a hydrophobic or water-fearing block, shown in orange. Nanoparticle formation is a phase of the nanodroplets, reorienting themselves so that the hydrophobic blocks are surrounded and stabilized by the hydrophilic blocks. This polymeric coating can be subsequently surface modified with various cancer-targeting ligands, such as antibodies or small molecules.
So now we are doing a delivery of nanoparticles.
The nanoparticle solution can then be modified into a form to allow for intravenous administration. The nanoparticles are quickly distributed throughout the body by the circulatory system, with which they are also delivered to the site of the tumor.
Red color one is the RBC, white color is the nanoparticles, and some of the white blood cells are there.
100 times smaller than red blood cells, nanoparticles possess the unique ability to permeate through the leaky walls of tumor vasculature, which are formed during the process of tumor growth.
That's why the tumor nanoparticle can progress through passive targeting way, slowly accumulating more and more.
Through this passive targeting technique, the nanoparticles can be concentrated within the tumor tissue.
Similarly, there can be done active targeting also.
On the cancer cell surface membrane, the nanoparticles encounter surface receptor molecules, microscopic markers expressed on cancer cells, but not expressed by normal tissues. The nanoparticles' targeting ligands bind specifically to these receptors, triggering a response also known as receptor-mediated endocytosis, which draws the nanoparticles into the cancer cell.
So like this way it can be delivered.
This process enables thousands of
Actually it start to release all the drug.
The anti-cancer drugs can be released in a controlled manner by the degradation of the polymer nanoparticle shell. The highly toxic chemotherapy can thus be delivered directly.
So that's the way we understand like how the it can be delivered. So now I have maybe another 5 minutes. I will try to say more about diagnostic and therapy together. That's a theranostic process. So the way we actually drinks like a tea, there is a catechin. So the one derivative of catechin and considering to herceptin together, they can form some nanoparticle which is more efficient to target to the tumor and which can be proved with time like up to 40 days. So this composite actually take over the tumor. So that's why the catechin can be effective also for some amount of treatment of cancer. Similarly, there is a magnetic nanoparticle. So now you can apply the magnetic field wherever the tumor location, if you know, then you can apply that particular place your magnetic field. Then you can control to reach the nanoparticle to that particular position. Now this nanoparticle you can make like a disc. So it's a very sharp in the edge. So now you applied high magnetic field, then start to vibrate it. Because of the vibration, actually they will try to cut the DNA. The DNA damage can be happen. So now here you can see because of the applied voltage, applied magnetic field. So now it starts to actually mechanical force is happening. And because it is sharp force, if it is happening at a cellular level, then all the cellular DNA also can the cancers. So similar way also we did like a one nanoparticle what conjugated with this DNA, and this DNA loaded with a drug molecule. It's now biocompatible. When we hit them, then they actually release the drug. By using this concept, we can consider to be a directly therapeutic as well as the diagnostic together. Here we can see it's a, we can visualize them. That means the diagnosis. At the same time, the therapy also works. With time, the cancer got treatment because of these nano formulations. So far, what we have seen different example. Now last, we can think about like what is the future of this nanoparticle. Like we are still in the 2025. So development of a new nanoparticles. Then from here it will be selection, and after selection, then there will be big animals like a dog, rabbits, monkey, and then it will come to the final phase as a clinical trial. That's around 2045, according to the Stephen-Whelan. So not only like these are that like a preclinical research. Obviously, question, does it anything comes in a commercial product? Yes, there is. And every year from 2005, 54, like a 2009, it's 2015, or even it is increasing more and more like a consumer products using nanomaterials are available. Major of them titanium oxide, zinc oxide nanoparticle, silver oxide, silica oxide nanoparticle. So these are the different commercial products. Majorly they are used for cosmetic purpose, like when you go for sun, for sunscreen, we are using this kind of different nanoparticle based materials. So here is the few example we can give it to you. Like we use the deodorant for antibacterial activity, then we use the toothpaste for whitening, then carbon nanotube for high energy transfer, like different kind of socks also used for antifungal, antibacterial activity. So that's why the nanoparticle have been used not only healthcare as well as in cosmetics and other home remedies. So we hope and we expect with future the number of nano product also will be more and more depending on their biocompatibility. So now we came at the end of my presentations. I think I'm on time. It's 2:55. So I would like to sincerely thanks to BC, Akal University, and HOD, Professor Sandeep Kumar, as well as the today's host and who invited me. And my sincere thanks to Dr. Shaha for giving me this opportunity, as well as DST sir for funding, NUS and Esther, and all the participants and all the faculties, those are here today. I wish that I could be offline and I can interact all of you directly. However, I did not get that opportunity, but still I sincerely thanks to all of you for listening. And I hope nobody's sleeping in the meantime. And I'm extremely happy if anybody has a question regarding this topic. Either you can directly ask me now or you can send me email. Thank you so much to all of you.
Thank you, Dr. Samantha. Indeed, it was a very good seminar presentation, and I can say that this is the just sequel of the previous seminar which Dr. Prakash has given regarding cancer in few months back. So I request students of our department to ask questions if you have, because I've already said that Professor Samantha, he will be happy to receive some queries from the students or maybe some other faculty members if they have any doubts regarding this session. Students, you can you can ask directly. If you cannot ask, then you can send me your questions in chat box. I will ask Dr. Samantha. Okay, if you don't have questions right now in your mind, okay, I think we got one question. Okay, Dr. Samantha, one question, one student from our MSc, she is asking when nanoparticles therapy is used, whether there is any side effect or not.
Okay, as I mentioned, the nanoparticle, if we consider bare nanoparticle, definitely there will be a highly side effect. However, the nanoparticle are modified with some biocompatible polymer or biopolymer, then it make a biocompatible, and usually it excreted from the body within the time, and then it does not have severe effect. However, for my personal experience, when I used eventually upconversion nanoparticle, when I keep it more than 2 months, I saw there was a animal, the mouse, their health is not that great like the normal one without treatment of upconversion nanoparticle. So depending on the nanoparticle, I can say some of the cases still toxicity will be a big concern. And some of the cases like a gold case, it's not much shown to be a that kind of toxicity. That's why it has been more extensively used towards gold nanoparticle rather having upconversion nanoparticle.
Okay, Dr. Samantha, we have another question. If we compare the different types of nanoparticles which are used for this therapy, what
Yes.
type of nanoparticles are more preferred? Like it is a just metal nanoparticle, metal oxide nanoparticle, or I can say that noble metal nanoparticle or non-noble metal nanoparticle, or is it metal oxide nanoparticles?
Okay, in that kind of cases, I will say now commercially available nanoparticles is a liposome-based drug carrier. Already it is available. That means organic nanoparticle is much more favorable with respect to inorganic nanoparticle, because whenever the inorganic particle will be there, it will be ultimately go to the cell. Then cell having the lysosome having like a more than 100 of enzymes, they start to actually degrade them. At the end, they should be excreted from the body. But because of its a metal, and if it is the concentration of metal is too high, there is a difficulty from excretion. But if it is an organic based nanoparticle, definitely they can degrade it much easier way, and as a metabolites, it can be excreted through urine or sweat or some way it can be excreted. So organic nanoparticle has much better advantage currently with respect to inorganic nanoparticles for final clinical application.
Okay, thank you, Dr. Samantha. There is another question regarding this presentation. Students wants to know that how much time it takes to carry out a study in any living cell.
Okay, living cell and animal. If we see about living cell, depending on the surface modifications of your nanoparticle. From my own experience, it will take 2 to maybe 12 hours, depending on that your cellular uptake happen. So minimum 2 hours can be good enough if your surface charge is positive. If the surface charge is negative, that is always difficulty, because our cell surface membrane is a kind of phosphate in the outer membrane, so which is negative repulsion does not allow them. So, and if we think about animal model, so usually we do the intravenous injections. So now I talk about the cellular level, 2 to 6 to 12 hours, and minimum we needed 2 hours. Then if we think about animal model, so during that time also, at least we needed 3 hours. Usually it goes to the liver also. And slowly, if we keep it longer time, we have studied up to 36 hours, 36 hours. Sorry, that is a 3 days. We continuously monitor it, and we have seen with time actually the tumor locations, the concentrations of nanoparticle, it is getting more and more, and it stay for even longer time. So that's why that's the timeline for in vitro as well as the in vivo.
Okay, there is another curious question, maybe silly to you, that when you do the study on a rat or a mice, how does they behave?
Yes, it's not silly question, because that should be the curiosity from the student point of view. I also, when I was first time, because being a chemist, I also very much curious about how to handle the animals. And what happened, there is some culture facility will be there. It has to be very much hygienic, and then you need to go with a proper dress code and you bring them. Then initially, like a rat, you cannot handle them without anesthetize them. So for the anesthetic purpose, there is a protocol. So when you go for OT, operation theater, there will be some anesthetic drug will be injected at the beginning, and then only whatever the operation we wanted to do, we can. In that kind of cases also here, if we wanted to do some imaging and other things during that time, we do anesthetized first, and then only we do any kind of imaging. But let's say if we wanted to just inject our nano composites, that time maybe sometimes without anesthetic it is possible. They have some special instrument, and this instrument we can like our mouse or our rat, we can push them to go there, and their tail will be outside. Now you can use your hand to finger to hold this kind of tail, and you need to find out where is the tail vein. So the vein you have to find it. So after that, you can actually the transparent solution, which or maybe colloidal, but should not have any kind of precipitation. Then because your blood vessels of the mouse will be very narrow. So then you can target to this vein and then you can slowly inject it. And if it is there is no particle, and if you are right vein you are injecting, you will feel like very smoothly can going inside. And after sometimes you can release the animal, and animal will be just normal. So they will not behave anything wrongly. And then but as it is already inserted, then it will circulate to the whole body and then slowly start accumulating to the right targeting area. That might be tumor.
Okay, thank you, Dr. Samantha. There are no more questions to ask. Maybe students are feeling a little bit shy to ask more questions, but students can send their questions to any of the teachers. They will mail to Dr. Animesh Samantha for answers, so that we can communicate in future also. And now I would like to take the opportunity to conclude this seminar on behalf of Department of Chemistry. First, I would like to thank our Vice Chancellor, Professor Gurmel Singh, for giving us this opportunity to conduct a webinar on recent topics of chemistry. This was our third seminar. Active supporter for this kind of program, and not only faculty members, our non-teaching staffs also. And not last but least of all, I would like to thank all the students of Department of Chemistry and other students of other departments who were present in this seminar to make it a successful. And I hope they have gained a proper information regarding the chemistry point of view or nanotechnology point of view to treat some very, I will say that this kind of disease are not common to us, and when it comes to any of us, we get seriously concerned. So in that point of view, what a chemist or what an nanotechnologist can do to help mankind, that information we have gained from this seminar. So thank you, Dr. Animesh Samantha, for your kind help and support in this seminar. And that's all from the Department of Chemistry. On behalf of Department of Chemistry, I like to thank all of you.
Thank you so much, Dr. Shaha, and really it's my great pleasure to get this opportunity, and thanks to all of you. Whoever have any question, you can send email, and Dr. Sandeep Saha already knows. If you by chance miss the email ID also, you can ask and send me email. Thank you so much to all of you. Probably last few words I'm unable to listen. Maybe internet connections. Okay, bye. Take care to all of you.
Thank you, Dr. Samantha.
Okay, now I can hear it. Yes. Okay, bye all of you. Take care.